So let's move on to objective 2, which is to describe the communication across chemical synapses and explain how neurotransmitters are released and then describe their actions after they are released. So if we think about the anatomy of a synapse, you want to think about the players that are involved and sort of thinking about that illustration that we just drew and thinking about sort of zooming into that axodendritic region. We want to think about what's happening in terms of that region of communication between the axon terminal and the postsynaptic neuron. And so zooming in there, we can really see the players that are involved in that synaptic transmission. So we have the presynaptic axon terminal, which is this region here. Okay, it's the terminal ending of the first neuron. We then have the neurotransmitter vesicles. So essentially these vesicles that exist within the axon terminal that have the important neurotransmitter stored in them. Now this neurotransmitter can be different things in different spots in the nervous system, so don't be too worried about what it is now, but the most abundant neurotransmitter in our body is acetylcholine, and we'll talk about acetylcholine coming up. Okay, for right now, we're just speaking about the players and their general role in this transmission. We can also think about the voltage-gated calcium channels. So we talked about before sort of all the locations of the various channels around our neuron. We said that the sodium channels had a concentration at the axon hillock and along the axon. We said that there was a concentration of calcium channels, right? Voltage-gated calcium channels at the axon terminal. And so these are the voltage-gated calcium channels that we are now looking at, okay? They have a high concentration here at the axon terminal. We also need to think about the cleft. So again, these two cells are not flush up against each other the way that electrical synapses happen. Instead, we have a bit of a distance, and this is called the synaptic cleft, right? Sort of this junction or region where there's a gap. We also want to think about the receptors. So there are receptors located on the postsynaptic neuron, right? The cleft of this postsynaptic or adjacent neuron. And these receptors are going to be specific to whatever the neurotransmitter that is stored in these synaptic vesicles are, okay? So this receptor has a specific binding site that matches whatever neurotransmitter is stored in the vesicles at the postsynaptic axon terminal. Excuse me, the presynaptic axon terminal. The other thing we want to think about are the enzymes that are present. So also along the postsynaptic membrane is the presence of a few important enzymes that play a role in that synaptic transmission. And then lastly, we have the reuptake molecules. So reuptake molecules are studied along the presynaptic membrane, so that presynaptic axon terminal. And these two structures, so the enzyme here on the postsynaptic membrane and the reuptake molecule on the presynaptic membrane, both of these structures function to reuptake or to clear the junction, clear the cleft of any extra neurotransmitter that would be lingering as a way to cease this communication or cease this signaling. So it's really important to have these two structures because they are going to be how we regulate this transmission, how we turn it on if there's the presence of that neurotransmitter in the cleft, but then how we turn it off by reuptaking this neurotransmitter or this enzyme, which is going to degrade the excess neurotransmitter. And that is how we're going to be able to regulate the signal or turn the signal off, okay? All right, so let's illustrate that here again as a way to add some more details. All right, so we're looking at the synapse here again, and I want you to make a note that we're only speaking about the chemical synapse, right? So from now going forward, we're only thinking about the chemical synapse, okay? So we've got our presynaptic neuron, right? Specifically our axon terminal here. And then we've got our adjacent cell. Okay, so really thinking about that, the dendrites of our, the dendrites of our postsynaptic neuron. So presynaptic neuron here. Postsynaptic neuron over here. All right, here we have our axon, right? This is our axon terminal. Okay, and so this region, this space here is called the synaptic cleft. All right, so the synaptic, let's put it here. This gap here is called the synaptic cleft. Okay. So let's put some of our players in on this presynaptic neuron. So along the length of our axon, we had these voltage-gated sodium channels, and this should be a bit of a review at this point, right? We've got the concentration of voltage-gated sodium channels. Really here along the gap. So if you think about this being myelin, right? When we talk about myelin insulating our neuron, we had the concentration of our voltage-gated sodium channels, which allowed for the initiation and propagation of the action potential. And so our action potential is leaping down the length of our neuron. So it's going from cell to cell. And then when it gets to the axon terminal, there are no longer voltage-gated sodium channels. So we can no longer have an action potential at the axon terminal. Instead, what we see are voltage-gated calcium channels. All right, so we've got these voltage-gated calcium channels that are studying our axon terminal here. All right. So this is a voltage-gated calcium channel. Oops. Okay. And so the next thing that happens is when the action potential gets to the voltage-gated calcium channels, it's actually going to depolarize them and they are going to open up. When they open up, they're going to allow for calcium to come rushing into the axon terminal. All right. We didn't speak much about the ECDF of calcium, but calcium has an ECDF such that when you open up those channels, they rush into the cell. All right. Okay, so that's kind of the first step of what takes place here. The action potential gets to the axon terminal. We open up these voltage-gated calcium channels. Calcium rushes into the cell, and then calcium is going to depolarize the axon terminal, right? It's going to become more positive in here. Calcium is positively charged, 2 times so. And depolarizing of the axon terminal actually triggers the release of our neurotransmitter vesicles. So we've got these vesicles that are inside our axon terminal, just being stored here, kind of just chilling out. And inside the vesicles, we have a specific neurotransmitter. All right. So when we depolarize this membrane, that triggers the dumping of these vesicles with this neurotransmitter out into the synaptic cleft. So our vesicles will now dump out into the synaptic cleft. Okay, so we can see all this neurotransmitter being dumped into the synaptic cleft. Okay, hope everyone's with me so far. All right, now this neurotransmitter is going to cross the synaptic cleft, right, cross this junction, and then it's going to bind to a very specific receptor. So let's draw this receptor here on our postsynaptic membrane. So we've got this receptor located on our postsynaptic membrane, and it is specific such that it will bind our neurotransmitter. okay? And if you remember us speaking about these ligand-gated channels, and we mentioned that a ligand is something that has to bind onto something in order to open it up, this is an example of the ligand-gated, so the ligand-gated channels. And so ligand-gated channels, which we said existed predominantly along the dendrites and soma, are the ones that will bind this neurotransmitter. All right, so let's make a note of that here. So we have the neurotransmitter, binding to our ligand-gated channel. And that is going to lead to our cellular response. Okay, and so essentially we go from action potential, which is electrical energy, right here, to neurotransmitter, which is chemical energy, right here. That neurotransmitter crosses the cleft, and then it binds to a ligand-gated channel, and this is how we retransmit that chemical energy back to electrical energy. It's going to help get us closer to firing another action potential on this adjacent neuron, right? On this postsynaptic neuron, okay? So this is what I mean by electrical to chemical, back to electrical, by the binding of this chemical, which is a neurotransmitter, to this ligand-gated channel, opening up this channel and facilitating the events, which would be either depolarizing or hyperpolarizing, depending on the state of this cell. And then that would potentially get us closer to our threshold and fire another action potential on the adjacent, on the postsynaptic neuron. All right. Now we will speak about two different types of cellular responses. So I want to put it into perspective here. We can think about a fast cellular response versus a slow cellular response. And so this is what we mean. This is where that is going to be taking place, just so you can have some context when we do go on to speak about the fast versus slow cellular responses. Now that is not it in terms of this transmission of signal here. The next thing that has to happen is we need to cease this signal, right? We don't want to have neurotransmitters being indefinitely dumped across the junction. We need to clear the junction of any excess neurotransmitter as a way to cease this signal or turn this signal off. And this is where our two molecules come into play. One is our enzyme here. So we've got this Pac-Man-like enzyme that is on the postsynaptic membrane. This is called acetylcholinesterase. Really long, fancy word. Acetylcholinesterase. And ASC meaning it's going to cleave or cut or degrade acetylcholine, right? So it's going to degrade the main neurotransmitter, which is acetylcholine. And again, don't be too worried about the names at this point or the types of neurotransmitters. This is just to give you the general sense of this signal, and we will add in some of the details later on, okay? But this enzyme is going to basically degrade any excess neurotransmitter. Oops. Okay, and then we also have another way to clear the junction. That is with the reuptake molecule. So we have a reuptake molecule that is on the membrane of our presynaptic neuron. So right here. And this is another way for us to clear the junction. So any type of extra or excess neurotransmitter can be reuptaken back into the axon terminal, and it can be recycled and repurposed into a new synaptic vesicle. Okay, so we have reuptake molecule. Oops, can't spell and type. Oh, can't spell and write. Reuptake molecule, which helps for us to clear the junction by recycling, repurposing some of that excess neurotransmitter and packaging it into a brand new synaptic vesicle. All right. And so these two things are working together, as I mentioned, as a way to clear the junction of excess neurotransmitter and cease that chemical signal. All right, I'm going to pause here and see what questions we have about this so far. And in the remaining minutes, we're just going to reiterate this with a bit of repetition just so that we can drive this home. You should be comfortable with the steps of this process. All right, you should be comfortable with the action potential, traveling down the axon, opening up this voltage-gated calcium channels at the axon terminal. Calcium rushes into the membrane. That triggers the exocytosis. In other words, the dumping of these vesicles, of the neurotransmitter that's stored in these vesicles into the synaptic cleft. Those neurotransmitter molecules binding to their relative receptor, that initiating the fast or slow response by the opening up of these ligand-gated channels, and then the clearing of the junction in order to cease the signal being done by the enzyme on the postsynaptic membrane, as well as the reuptake molecule on the presynaptic membrane. All right.
I had a question about the reuptake molecule and the enzyme. So does it matter which of the, like, sorry,
the neurotransmitter
neurons go to which side?
Say again, sorry.
Does it matter which neurons go to like the reuptake model versus the acid?
Great question. Yes, it does. So the reuptake molecule is always on the presynaptic side. So the side that has the vesicles is where we want to repurpose that neurotransmitter in terms of creating new vesicles. The enzyme is always on the postsynaptic side. Okay, I think that was your question. I hope I answered that.
Okay, so if you know, they're on the right sides and everything, is there like certain ones that are reused too much, or is there no such thing as reusing them too
much? There's no such thing as reusing them too much. There are a few diseases that we'll speak about where they're not present in enough quantities. And so like Myasthenia Gravis is a really interesting example where we can have not enough of our neurotransmitter here. And so having more of these things blocking these things would help to allow for that neurotransmitter to linger at the junction for longer or for longer periods. So there's no such thing as reuptaking it too much, but we do have instances where there's not enough neurotransmitter, and by blocking these, we can allow that neurotransmitter to linger at the junction for a longer period of time. And myasthenia gravis is an example that we will speak quite a bit about as a clinical example. Right, great questions. Any other questions? Any other questions here? Okay. Now we'll add some more detail in terms of the reuptake molecule, but there are several different ways that we can reuptake or degrade this neurotransmitter, and we'll talk about that as we kind of go on. All right. Okay, so here we're just kind of going over the steps of these events. Again, just repetition at this point. So our action potential gets down to the axon terminal. It opens up our voltage-gated calcium channels. This triggers the exocytosis. So exocytosis is one of those transport processes that we talked about previously, where the vesicle membrane fuses with the synaptic membrane, and then the neurotransmitter can be sort of evacuated or dumped into the cleft. It's then going to cross the cleft, bind to its very specific receptor, and help to bring about the response on the adjacent cell. At that point, we need to clear the junction in order to cease this signal. And so we can do so by degrading the excess neurotransmitter via the enzyme that's present on the postsynaptic side, or we can degrade or recycle the neurotransmitter as well. So we can also recycle this neurotransmitter in its whole state. We can clear this neurotransmitter, so it can simply just diffuse away from the cleft. or it can be degraded and then diffuse away from the cleft in that way. Okay, so let me say that again. We can reuptake whole neurotransmitter molecules as is. We can degrade them down to smaller pieces. And then from those smaller pieces, they can simply diffuse away, or they can diffuse away in their whole state as well. Okay, so there are a couple of different options for clearing the junction, but they mainly rely on these two molecules. So the enzyme on the postsynaptic side and the reuptake molecule on the presynaptic side. Sort of the main thing to point out is that once we degrade it, it can no longer be reuptaken. So it has to be reuptaken in its whole form. Otherwise, it can diffuse away in its whole form, or it can be degraded and then diffuse away. All right. Now we want to talk about the 0.5 to 5 millisecond delay, which exists between the time that the action potential arrives at the axon terminal and the time that it takes for us to then see some type of change in the membrane potential on our postsynaptic cell. And the reason that there's a delay for that is because it takes time for exocytosis to occur. So it takes a bit of time for the docking of these vesicles to the membrane, and then the exocytosis, so the dumping of the membrane, dumping of the neurotransmitter out of the vesicle and into the cleft as the membrane of the vesicle fuses with the membrane of the axon terminal. Okay, and so that is what takes up that time. That's what causes that 0.5 to 5 millisecond delay, and kind of what slows this whole thing down, right? We talked about the idea that electrical synapses are really rapid. The slowing down of chemical synapses is the time that it takes for this chemical inside this vesicle to be dumped into the cleft, because we have to merge the membrane of the vesicle with the membrane of the axon terminal. And all of that just kind of adds some time, some kind of, some delay in the process. And this is why it takes a bit more time. Now, what this delay is not due to is diffusion across the cleft, okay? We would kind of be tempted to think, oh, it'll take some time for this neurotransmitter to diffuse across the cleft. That is not true. So the delay that we see between the arrival of the action potential and the response, the membrane potential change on our postsynaptic neuron, is not at all due or not at all owed to the time that it takes for diffusion to occur. Diffusion occurs pretty rapidly, but what it is due to is the exocytosis process, the docking of the vesicle, the fusion of the vesicle membrane, and then the dumping of that neurotransmitter. Okay, that is where we see that delay.